Hello Sanjay, very good evening. Thank you for joining. See, today we are going to discuss is about biological classification. So shall we start Sanjay? Am I audible clearly? Good evening Anita. Good evening Sanjay. Am I audible pa? Am I audible to you? All? Yes. Thank you Sanjay for your answer. Yes Dharni, very good evening. So let us discuss is about biological classification. Very important chapter for NEET as we get 4 questions from this. So you all know what is classification. Classification means grouping of organism. So நம்ம இங்க என்ன பண்றோம்? We will group it. Okay. So grouping of organism. So who first made the classification or grouping of organism? யார் முதல்ல grouping of organism கொண்டு வந்தாங்க அப்படின்னு பார்த்தோம்னா it is Aristotle. So we call him as ancient father of classification. ஏன் அவரை ancient father of classification அப்படின்னு சொல்றோம் அப்படின்னு பார்த்தோம்னா அவர் தான் முதல்ல he started that Plantae and Animalia அப்படின்னு ஒரு குரூப்பிங்க கொண்டு வர்றார். So Plantae அதுல என்னெல்லாம் கொண்டு வந்தாரு அப்படின்னு நீங்க பாத்தீங்கன்னா herb, shrub and trees. ஸோ இதுதான் வந்து அவர் கொண்டு வந்த குரூப்பிங். அதுக்கப்புறம் நீங்க பாத்தீங்கன்னா அனிமாலியே. ஸோ அனிமாலியில பார்த்தோம்னா அஃப்கோர்ஸ் வித் பிளட் அண்ட் வித்தவுட் பிளட், அனீமியா எனீமியா அப்படின்னு நம்ம கிளாஸிஃபை பண்றோம். அனீமியா அண்ட் எனீமியா. ஸோ ஐ ஷோ யூ சில்ட்ரன். இதுவரை நம்ம பார்த்தது என்ன அப்படின்னு பார்த்தோம்னா அஃப்கோர்ஸ் இதுல வந்து நம்ம கிளாசிபிகேஷன் ஹூ இஸ் தி ஏன்ஷியன்ட் ஃபாதர் அப்படின்றத நமக்கு கேட்கலாம். So, இது அரிஸ்டாட்டில். அஃப்கோர்ஸ் பிளான்டேவை வந்து ட்ரீ, ஷர்ப் ஆஸ் வெல் அஸ் ஹர்ப்ஸா கொண்டு வராரு. அதுக்கப்புறம் அனிமல்ஸ்ல பாத்தீங்கன்னா வித் பிளட் அண்ட் வித்தவுட் பிளட் அப்படின்னு கிளாஸிஃபை பண்றாரு. ஸோ வித் பிளட் அப்படின்னு பார்க்கும்போது தே ஆர் மேமல்ஸ், லிசர்ட்ஸ், பேர்ட்ஸ் அண்ட் ஃபிஷஸ். வித்தவுட் பிளட் அப்படின்னு பார்க்கும்போது ஹார்ட் பாடிஸ் அண்ட் சாஃப்ட் பாடி அனிமல். இப்போ ஹார்ட் பாடிக்கு எக்ஸாம்பிள் என்ன கொண்டு வந்தோம் நம்ம? அஃப்கோர்ஸ் இன்செக்ட்ஸ். சாஃப்ட் பாடியில நம்ம என்ன கொண்டு வரோம் அப்படின்னு பார்த்தீங்கன்னா ஷெல் அண்ட் நோ ஷெல். அஃப்கோர்ஸ் ஷெல்லுக்கு வந்து ஷெல் ஃபிஷ், நோ ஷெல்க்கு ஜெல்லி ஃபிஷ். ஸோ இஸ் அவர் ஏன்ஷியன்ட் ஃபாதர் அப்படின்னு நம்ம வச்சுக்கலாம். இப்போ அடுத்து நம்ம பார்க்க போறது என்ன அப்படின்னு பார்த்தீங்கன்னா மாடர்ன் ஃபாதர். இப்போ மாடர்ன் ஃபாதர் யாரு? யாராச்சும் சொல்ல முடியுமா? எஸ் கீர்த்தி, வெரி குட் ஈவினிங். மாடர்ன் ஃபாதர் அப்படின்னு பார்த்தோம்னா இஸ் அவர் லினேயஸ். ஓகேயா? அப்ப லினேயஸ் என்ன கொண்டு வராரு? லினேயஸ் ஆல்சோ மேட் 2 கிங்டம் கிளாசிபிகேஷன். சோ இந்த 2 கிங்டம்ல நம்ம என்ன கொண்டு வரோம்? பிளான்டே அண்ட் அனிமாலியே. அஃப்கோர்ஸ் உங்க எல்லாருக்கும் தெரிஞ்சதுதான். இஸ் வெல் நோன் பர்சன் ஃபார் பைனாமியல் நாமென்க்ளேச்சருக்கு கொஸ்டினை கேட்கலாம் லினேயஸ். மாடர்ன் ஃபாதர் ஆல்சோ லினேயஸ் ஒன்லி. சரிங்களா? இப்போ இந்த two kingdomல plant is it accepted in the kingdom classification of Linnaeus வந்து is got accepted? Can anybody answer me? It was not accepted. Am I correct? Of course. என்னெல்லாம் அவரோட ப்ராப்ளமா இருந்தது அப்படின்னு பார்த்தோம்னா அஃப்கோர்ஸ் அதுல வந்து என்னெல்லாம் ப்ராப்ளமா இருக்கு அப்படின்னு பாத்தீங்கன்னா ஆஸ் யூ நோ அவரு யூனிசெல்லுலார் அண்ட் மல்டி செல்லுலார கிளாஸிஃபை பண்ணல இல்லீங்களா? இப்ப பாத்தீங்கன்னா நமக்கு என்சிஆர்டி லைன் என்ன சொல்லுது? கிளாமிடோமோனாஸையும் ஸ்பைரோகைராவையும் ஒண்ணா வச்சிருப்பார். எஸ், தாரணி வெரி கரெக்ட் ஆன்சர். சோ கிளாமிடோமோனாஸ் வந்து உங்களுக்கு தெரியும், அது யூனிசெல்லுலர். அண்ட் ஸ்பைரோகைரா அது மல்டி செல்லுலர். ஸோ இது எல்லாமே ஒன்னாவே நமக்கு கன்சிடர் பண்ணியிருப்பாரு அவர். அதுக்கப்புறமா பாத்தீங்கன்னா அஃப்கோர்ஸ் ரெண்டாவது பாத்தீங்கன்னா போட்டோசிந்தட்டிக் நான் போட்டோசிந்தட்டிக் இன் த சேம் குரூப். போட்டோசிந்தட்டிக் எது? அஃப்கோர்ஸ் பிளான்ட்ஸ். நான் போட்டோசிந்தட்டிக் எது? ஃபங்கை. இதெல்லாம் ஒரே குரூப்பா இருப்பாங்க இல்லீங்களா? ஸோ தட் ஆல்சோ இஸ் நாட் accepted actually. Non photosynthetic, that is plants and fungi. And then you know, of course, prokaryotes and eukaryotes together. இப்போ பாக்டீரியாவும் பிளான்ட் எல்லாம் ஒண்ணா வருது இல்லீங்களா? prokaryotes versus eukaryotes. So that was also not accepted. So like this it was controversial அவரோட. You know euglena அப்ப நம்ம எங்க வைப்போம்? Euglena, if you see, it is having plant character also, animal character also. But he kept it in plant. Yes, Ajay, very good evening. Thank you for joining, Ajay. Yes, my dear friend Pushpa Kumar, thank you for joining and supporting. Euglena, of course, இது பிளான்டோட கேரக்டரும் இருக்கு அண்ட் அனிமலோட கேரக்டரும் இருக்கு இல்லையா? அப்போ அதை வந்து நம்ம எங்க வைக்க முடியாது? பிளான்ட்ல வைக்க முடியாது, அனிமல்லயும் வைக்க முடியாது அப்படின்னு ஒரு குரூப் ஆஃப் பீப்புள் வேர் ஆர்கியூவிங். அப்படிதான் இந்த பயாலஜிக்கல் கிளாசிபிகேஷன் அண்ட் ஸ்டார்டட். தென் தெர் ஆர் சோ மெனி கான்ட்ரவர்ஷியல். அதுக்கப்புறம் உங்களுக்கு 3 கிங்டம் வந்தது, 4 கிங்டம் வந்தது. ஃபைனலி அக்செப்டட் மாடல் என்ன அப்படின்னு பாத்தீங்கன்னா 5 கிங்டம் கான்செப்ட். Who proposed 5 kingdom concept? யாரானு பதில் சொல்ல முடியுமா? 5 kingdom concept is proposed by you all know the answer. Very good, Dharini. Yes, thank you. R.H. Whittaker is the correct answer. Okay. And which year he proposed that 5 kingdom concept is a possible question. Of course, it is 1969 he proposed. Okay, அப்ப அவர் என்னெல்லாம் கேரக்டர் எடுத்துட்டாரு? Based on that statement, a question will come. So, what are the characters he has taken for classification? Number 1, of course, it is cell structure, thallus organization, nutrition, reproduction, and also phylogeny. So phylogeny, you know, evolution is also considered in 5 kingdom classification. Fine. What he didn't consider for classification? If you remember the page of the biological classification in NCERT book, children, அங்க நமக்கு ஒரு paragraph கொடுத்திருப்பாங்க, ஒரு tabular column இல்லையா? What is the animal character of euglena? If you put euglena in dark kanna, it will enter into holozoic mode of nutrition. ஓகே, அதுக்கு 2 ஃபிளாஜெல்லா இருக்கும் இல்லையா? ஒரு சின்ன ஃபிளாஜெல்லா, ஒரு பெரிய ஃபிளாஜெல்லா. அந்த சின்ன ஃபிளாஜெல்லா வச்சு அது ஒரு சின்ன வேர்ல்பூல் கிரியேட் பண்ணி என்கல்ஃப் பண்ணும் நியூ ஃபுட். So it cannot prepare when it is in dark. அதுக்கு நிறைய கேரக்டர் இருக்கு. இன்னொன்று வந்து பார்த்தீங்கன்னா ஆஸ்ட்ரோசாந்தின் அப்படின்னு ஒரு ஐ பிகமெண்ட் கூட ஃபார்ம் ஆகும் டார்க்ல போடும்போது. அண்ட் குளோரோபிளாஸ்ட் எல்லாம் காணா போயிடும். அதனால அது அனிமல் கேரக்டரிஸ்டிக் ஃபீச்சர் இன் யூக்ளினா. ஓகேவா தாரணி? ஃபைன். இப்போ வாட் இஸ் நாட் கன்சிடர்டு பை விட்டாக்கர் ஃபார் ஃபைவ் கிங்டம் கிளாசிபிகேஷன்? யா. ஸோ எது வந்து நம்மளுக்கு ஃபைவ் கிங்டம் கிளாசிபிகேஷன்ல விட்டேக்கர் எடுத்துக்காத ஒரு கேரக்டர்? இட் ஸ்டார்ட்ஸ் வித் என். ஐ வில் கிவ் யூ அ க்ளூ. எண்ணில் ஆரம்பிக்கும். எஸ் திலக், வெரி குட் ஈவினிங். Is he didn't mention about nuclear membrane. சரியா? அந்த டேப்லெட் காலம்ல வந்து உங்களுக்கு நியூக்ளியர் மெம்பரேன் கொடுத்திருப்பாங்க. பட் விட்டாக்கரோட எக்ஸ்பிளனேஷன்ல பாத்தீங்கன்னா only these five will be there. That is cell structure, thallus organization, nutrition, reproduction, phylogeny. Nuclear membrane present and absent, it is mentioned only in the tabular column, but it was not considered by Whittaker. அது நல்ல ஞாபகம் வச்சுக்கணும். ஸ்டேட்மெண்ட் டைப் கொஸ்டின்ல நமக்கு அது பாசிபிளா கேட்பாங்க. I hope you are clear now. Any doubts? Shall I proceed? So as we discussed this Aristotle's picture, of course you can see this is Linnaeus who made two kingdom classification. So Linnaeus made a two kingdom classification, which is kingdom Plantae and kingdom Animaliae. நம்ம டிஸ்கஸ் பண்ணோம் இல்லைங்களா? இதுல வந்து of course he kept everything together. Prokaryotes, eukaryotes ஒண்ணா வச்சுட்டாரு. யூனிசெல்லுலார் மல்டிசெல்லுலார் ஒண்ணா வச்சுட்டாரு. and he didn't differentiate எது வந்து autotroph, எது வந்து non photosynthetic, that is heterotroph, that he didn't differentiate. Yes sir? Of course, he is Whittaker on 1969 who made this five kingdom classification based on cell structure, thallus organization, mode of nutrition, reproduction, and phylogeny relationship. Yes, this is Whittaker, and of course, the five kingdoms are here. You can see the Monera and kingdom Protista, fungi, Plantae, as well as Animaliae. Okay, children. Fine. So I told you what are the possible questions, which year he published, and of course what was not considered. If you see the tabular column, nuclear membrane is mentioned. Okay, but it is not proposed by Whittaker. That we made actually. Now can anybody answer me? How many of the five kingdom are parasitic? How many of the five kingdom are parasitic? Is it all five, 4, 3, 2 or 1? How many of them are parasitic? Yes. Can anybody answer me? How many out of five kingdom are parasitic? The answer is all five. Okay, because Monera bacteria or parasites. Protista, we have amoeba, parasitic. Plantae, Cuscuta is parasitic. Fungi are parasites, in animals we have tapeworm or ringworm, sorry, not ringworm, roundworm. All these are parasites. Yes. Parthasarathi, very good evening. So, all five would be the parasitic. Thank you for answering, Parthasarathi. Yes. Now, let me ask you, how many are autotrophic? How many are autotrophic? Out of the five kingdoms, how many are autotrophic? Can anybody answer me? Is it five or four or three or two or one? Very good, Parthasarathi again. Yes, it is three. Excellent. Bacteria, cyanobacteria will do photosynthetic characteristic feature is there, of course. And then you are having plantae, very well it will do the photosynthesis autotrophic. And then in Protista, we have chrysophytes will do photosynthesis. Dinoflagellates will have chlorophyll. Even Euglena will have the chlorophyll. Yes, Dharini and Tilak, thank you. It is three. Will do autotrophic nutrition. Okay. So it is bacteria, plantae and protista will do autotrophism except fungi and animals. Okay, pa. Fine. So this is regarding the tabular column. Then next one we are going to discuss about Monera. So the first kingdom where includes the bacteria, Arachidacteria. So it includes both Eubacteria and Arachidacteria. Fine. Now let us see where is the habitat for these bacteria. Where do we find the bacteria? It is cosmopolitan. Am I correct? Cosmopolitan means we found everywhere. You can't say this is not having any bacteria except a sterile zone. Okay. So like it is cosmopolitan in distribution. And we know that it also have different shapes. Yes, Tilak, very good. It is everywhere we find. And can anybody answer me? What is that shape when it is spherical in shape? The bacteria are named as what? Yes, Tilak, that is for you. Mm, the bacteria which are round in shape are called dash. Is it cocci? Is it vibrio? Very good, Dharini. Excellent. It is cocci. Which is comma shape called which is comma shape called yes, very correct. This is for you, Tilak Parthasarathi. Yes, Sanjay. Yes, yes, that is for you. Yeah. Now, what is comma shape bacteria are called? Very good, Vibrio. 100% correct. Yes, yes, Vibrio is the correct answer. Okay, and spiral shape is Yes, Sanjay, very correct. And Dharani, you are also correct. Yes, Pa. Now, spiral shape is very good. Spirilium, 100% correct. Spirilium. Okay. And then rod shape is rod shape is yes, Shubhan. Very good evening, Shubhan. Yes, Shubhan, this is for you. See, we are discussing actually biological classification. We just started Monira, Shubhan. Okay. And rod shape, as you know, basil is very correct. Dharini, 100% correct. Yes, it is bacilli. So, based on this, questions will come. Comma shape is what? Spiral shape. Or at least in statement type question, you can expect this. Okay. Now, we know habitat as well as where it is present. Now, based on the structure, the very common question is metabolic diversity. You can find among the five kingdom which one? Very commonly. Metabolic diversity we found in which organism or which kingdom? Metabolic diversity. Yes, Parthasarathi, very correct. 100%. Yes. Yes, Dharini, very correct. Okay, so metabolic diversity we found in Monera because it is showing photosynthetic autotrophic, chemoautotrophic as well as heterotrophic. Okay, so it is autotrophic, heterotrophic, autotrophic in the sense both photoautotrophic and chemoautotrophic. So, you know, it is metabolic diversity is a very common question. Autotrophic, chemoautotrophic, as well as you can find the heterotrophic. So, let us see. Kingdom Monera. So, in Kingdom Monera, you can find the bacteria everywhere. You can find even on the desert or even on the snow, deep oceans, in our hand also. Everywhere we can commonly find bacteria, which is the most abundant organism. Yes, can you all see the organism bacteria? Various shapes, of course, everywhere you can find, and some are parasites also. Even in soil, you can find bacteria, or in deep oceans, snow, deserts, everywhere. Now, this we discuss various shapes, of course, spherical shape, rod shape, comma shape, spiralium shape. So, all these are different shapes of bacteria which we discussed already. And this is the structure of bacteria we are going to discuss. Yes. If you see the bacteria, you can find the capsule. So capsule is gelatinous in nature. It may be slimy, it may be rigid also. I hope you remember, based on the slimy and rigid, Griffith did transformation experiment. Cell wall. This is cell wall which is lined in yellow color. Cell wall. Can anybody answer me? The cell wall of bacteria is made up of what? So cell wall of bacteria is made up of what? Yes, Tilak, glycocalyx is outermost. Capsule is made up of glycocalyx. And cell wall is made up of uh, yes, Parthasarathi, of course, lipids you find in plasma membrane. Is it not peptidoglycan, Kanna? Peptidoglycan, possible question, right? Peptidoglycans. The cell wall of the bacteria is made up of peptidoglycans. Of course, based on the cell wall composition, we can even classify gram positive or gram negative. If they have teichoic acid, we call them gram positive. They take up the stain and gives violet color. Yeah. And if at all teichoic acid is not there and it may be gram negative, it does not take up the violet color and it will be pink in color. Now this is plasma membrane, of course. The plasma membrane showing foldings. You remember what is the name of the folding, children? Start with M. See, I gave some fill-ups here. Yes, Parthasarathi. Very correct. The foldings of the plasma membrane. Very good, Dharini. Excellent, Dharini. Yes, 100% correct. Yes, it is mesosomes. Okay, based on this, many possible questions are there, pa. Mesosomes. So, what are the functions of mesosome? Yes, Parthasarathi. Very correct. So mesosomes will do DNA synthesis and then do DNA distribution, cell plate formation, that is cell wall formation, and then respiration. As you know, non-membrane bound organelle alone present in the bacteria. So all other functions are carried out only by mesosome. Yes. Now, what is the only cell organelle you find in the bacteria? What is the only cell organelle you find in the bacteria? Yes, respiration. Very correct, Parthasarathi. The only cell organelle we found in the bacteria, which is non-membrane, is dash. Very good, Dharini. Excellent. It is correct. Ribosome is the correct answer. You can see even in the slide, it is ribosome. And that is 70S ribosome. And you know, ribosomes are the protein factories. When many ribosomes arrange like a chain in polypeptide synthesis, it is called water. When many ribosomes arrange like a chain, it is called many ribosome. Can you see the picture? Many ribosome arranged like a chain. It is called what? Very good, Dharini. Excellent. Again, it is polyribosome or polysome. Possible question. Polysome or polyribosome. Okay. Fine. Then you can see these are pili. Pili are for conjugation. And to the harder substances like a rock, the bacteria will get attached using this. It starts with F. Can anybody guess? Okay, give two words. Like a rock or any harder substance, the bacteria will get attached only using this. Shall I give the next word? It is fimbrium. Okay, so fimbria is the one which is for attachment. Then you can see the other structure is flagellum is used for locomotion. Of course, it is made up of a protein called flagellin. Okay, and you know what is the genetic material? Is it double stranded DNA or single strand DNA? Is it double strand DNA or single strand DNA? What is the genetic material of bacteria? Yes, my friend Pushpa Kumar. Any guess? Is it double strand or single strand? The genetic material is yes, Shubhan, very correct. It is double strand DNA is the correct answer. Okay, and it is otherwise called possible question. It is otherwise called nucleoid or genome. Okay, so it is otherwise called nucleoid or genome. Yes, Parthasarathi and Tilak, very correct answer, 100% correct. It is double standard and it is otherwise called nucleoid or genome. Okay, like that, you have a circular extra chromosomal DNA also found here. That extra chromosomal DNA is called dash. The extra chromosomal DNA of bacteria is called a dash. It starts with P, end with D. The extra chromosomal DNA, which is circular and double stranded. Very good, Parthasarathi. Excellent. Yes. So it is plasmid. Very correct answer. The extra chromosomal circular DNA is plasmid. So I discuss all possible questions of the bacteria here. And you know, bacteria is divided into two criteria where we have yes, Dharini, very correct answer. It is plasmid. Okay. You can find two types of bacteria. One is Araki bacteria, another one is eubacteria. Of course, we discussed they are having metabolic diversity, photosynthetic, chemosynthetic, parasitic, saprophytic. All sort of nutrition you can find. Now, Araki bacteria, halophiles, Dermoacidophiles, methanogens. You know very well, methanogens commonly where do we find methanogens? Like methanococcus, methanophilus. All these where do we commonly find? Very good, Parthasarathi. Very correct. Yes, Dharani. Yes, very correct. Gut of ruminants is the correct answer. Where we find commonly in the ruminants. Thermoacidophilic, of course, hot vent. All these area you can find. Of course, you know, Thermus aquaticus, example. Halophiles in lagoons. We can find it in lagoons. Okay, so this is regarding Arachidactylia. Now, can you tell me what is the difference between Arachidactylia and Eubacteria mainly? The main difference between Arachi and very good. Yes, yes, excellent, excellent. Yes, Pa. Yes, Pa Dharani. And Parthasarathi, yes. They have special cell wall structure to protect extreme habitats. Only they can survive in extreme habitat. As you know, they have side lipid chain. Um, but NCRT line says they have special cell wall structure. Okay. What is that special cell wall structure? They have side lipid chain additionally present. So that is why they can able to tolerate a very harsh climate even. Okay. Thank you. Very good, Dharini and Parthasarathi. Excellent answering. Okay. So you can see the examples here, methanogens in the gut of ruminants, thermoacidophiles in the hot vents, halophiles in the lagoons. So this is the picture shows halophiles, thermoacidophiles and methanogens and their habitat. This is regarding the Arachidactyria. So the possible question is, so different cell wall structure, exactly NCRT line. Make them survive in extreme condition. So here you can see the bacteria. They have a cell wall and flagella for locomotion. Of course. Now we are proceeding to the cyanobacteria. So the cyanobacteria you can find where is the habitat? Of course, freshwater. First possible question. And they bloom well in, they very commonly bloom well in polluted water. So we find abundance in the polluted water. Okay. So they are unicellular and filamentous structure. So you can find they bloom in polluted water. Of course. They are covered by a gelatinous sheath. You can find a gelatinous sheath. Okay. And you know very well they are involved in nitrogen fixation. What is the structure involved in nitrogen fixation? The structure involved in nitrogen fixation is what? Very good, Dharini. Excellent. Yes, Partha Saradi. Very correct. Yes, excellent answer. Heterocyst. Possible question. Okay. So, you know, heterocyst they will be having which helps in fixing the nitrogen. The next possible question, of course, it fixes the nitrogen in three forms. What are they? What are the forms it fixes the nitrogen? The forms it fixes nitrogen. What are the forms it fixes the nitrogen? So it fixes in the form of, very good Tilak, excellent, excellent. Yes. Nitrates, nitrites and ammonia. So nitrates, nitrites, ammonia. So these are the forms. Yes, Partha very correct. Nitrates and nitrites, ammonia. Yes. Heterocyst is very correct answer, Shiva. Excellent answering. Okay. So these three, they fixes the nitrogen. Okay. Now, apart from this, the chemosynthetic autotrophs may also involved in fixing four another minerals. What are those four minerals? Can anybody answer me? Yes, Dharini, Parthasarathi. So what are the minerals it fixes? Chemosynthetic bacteria. Nitrogen. Very good. Excellent, Tilak. Excellent. Yes. Nitrogen. Phosphorus. Yes, Kirti. Yes, Parthasarthi. And yes, Shiva. Excellent answering. Yes, nitrogen, phosphorus, sulfur and iron. Okay, so these are the four actually involved in fixing the nitrogen. So nitrogen, phosphorus, sulfur, iron, the last line of NCRT in that cyanobacteria. And one more possible question, children. Name the pigment found in cyanobacteria similar to plant. Pigment found in cyanobacteria similar to plant. Is it chlorophyll A or chlorophyll B or chlorophyll C? What is the pigment? Yes, Tilak, excellent answering. Chlorophyll A is the correct answer. Yes, chlorophyll A only. Okay, so that is another possible question. So I discuss all possible question. Where it is bloom in polluted water. Of course, the structure fix the nitrogen heterosis. It fixes the nitrogen in the form of nitrates, nitrites and ammonia. It also fixes nitrogen, phosphorus, sulfur and iron. Okay, and it has a pigment chlorophyll A similar to that of the plant. Yes, Pushpa Kumar. Very correct answer. Yes, Parthasarathi, very correct answer. Chlorophyll A. Okay. So it fixes nitrogen in the form of nitrates, nitrites, ammonia we discussed. Of course, it also fixes recycling, nitrogen, phosphorus, iron and sulfur. Yes. And bacteria are known as heterotrophic because they are most abundant. Of course, you can't count them. In previous chapter, we said biodiversity, the organism is 1.7 to 1.8 and all. Here you cannot count. These are how many number. You cannot count it. Na? So they are abundant in nature. They are mainly decomposers. So what are the purpose? They are used for making curd. You can see curd making lactobacilli. Okay. So lactobacilli is involved in curd making, antibiotic erythromycin, streptomycin, tetracycline. All these are antibiotic. Fixing nitrogen. Just now we discussed fixing nitrogen. Yes, Shiva, very correct answer. Okay. And some are pathogen even causing disease. See this question. Which among the following is for bacterial disease? These four. Vibrio cholerae, Salmonella typhi, tetanus, citrus canker in plants causes the diseases. Are you all clear, children? About Monera, we discussed first kingdom. Any doubt? And bacteria reproduce favorable condition by fission. Unfavorable condition, they produce water, spore. So unfavorable condition, spore is produced. And also reproduced by sexual reproduction, which is also called genetic recombination. It may be transformation, transduction, or conjugation. Yes, sir? Are you all clear? Fine. So this is mycoplasma. Next one we are going to discuss is about mycoplasma. Yes, Kirti, thank you. So mycoplasma you can find without cell wall. So cell wall is not there. Okay. Anaerobic. So it can survive without oxygen also. And smallest living cell. Of course, it is pathogenic to plant and animal. It causes pneumonia. So only it is even called a PPLO, pleuropneumonio-like organism. Or otherwise, they are even called jokers of microbiology. Yes, sir. So we finished actually Kingdom Monera with that. Any doubt, children? Yes, Sanjay, Parthasarthi, Pushpakumar, Shiva, Kirti, Dharini, Anita. Anybody? Yes, thank you, Shubha. Okay, so next one we are proceeding to the Kingdom Protista. So Kingdom Protista is the one which has link with the Plantae, Animaliae and fungi. Okay. Yes, thank you, Dharani. Thank you, Parthasarathi. Okay. As you know, they are having a link with plantae, animalia, and fungi. And you know, they are all unicellular and eukaryotes. So, how they are considered as eukaryotes? They have true nucleus and other membrane bound cell organelle. Okay. Can anybody tell me whether they reproduce sexually or asexually or both? Whether the protista reproduce asexually or sexually or both? Yes, Tilak, very correct. Yes, very correct answer. Both is the correct answer. It of course reproduce asexually and sexually by zygote formation and cell fusion is the correct answer. So you can see it's a single cell eukaryotes. You can find nucleus and membrane bound organelles. They have either flagella or cilia for locomotion. So you know it is having a link with plant, animal and fungi. They are aquatic mostly. They reproduce asexually, sexually by cell fusion and zygote formation. Yes, Shiva, thank you. Fine. So these are some of the protista. Chrysophytes, dinoflagellates, euglenoid, slime mold and protozoan. So how are we going to learn? We are going to learn based on their habitat and unique features as well as their mode of nutrition, reproduction. So first one, chrysophytes. What is the common name of chrysophytes? What is the common name of chrysophytes? Yes, very good, Dharani. Very good. It is diatoms or desmids or golden algae. Diatoms, desmids or golden algae. So these are other names. What is the habitat? Where do we find a habit of these algae? Yes, they are mostly aquatic and freshwater. Some are marine. So off you can say. Yes, Parthasarthi. Okay. So you can find mostly aquatic is the NCRT line. Okay. And some are marine, of course, as you said. Yeah, freshwater as well as marine is most correct answer, Tilak. Excellent answering. So some are freshwater and some are marine. Okay. And you know, the cell wall is made up of the cell wall is made up of. Yes, very good. Correct answer. Silica. Excellent answer. So, it is like a soap box. Okay. So, outer layer we call it as epitheca. Inner layer we call it as hypotheca. And they are made up of silica. Okay. So, it may be like a box or it may be centric nature also. Yes. Kirti, very correct answer. And Parthasarthi. It is silica. So it cannot be decomposed and it forms diatomaceous earth. Yes or not? So it cannot be decomposed, it forms diatomaceous earths. Okay, what are the uses of these silica? Uses of silica. Very good. Yes, it is used in filtering and polishing. Okay. So all these, yes, very correct, Kirti. Okay. So it is used in filtering and polishing. All these usage is there in diatomaceous silica. Of course, who are the chief producer of ocean? For this question, what would be the answer? Chief producer of ocean. Yes, Subhan, very correct. Diatoms is the correct answer. Possible question. Okay. Yes, Dhanani, chrysophyte or diatom is the correct answer. Okay, so it is having silicaceous cell wall and they are the chief producers in the ocean. Very possible question. And they undergo either asexual reproduction or sexual reproduction and they produce oxospore also. And from that sexually reproduce again they form the diatoms. So you can see some of the diatoms here. As it is golden in color, it is also called jewel of plant kingdom. So it is even called jewel of plant kingdom. Yes, Parthasarthi, very correct answer. So these are the possible questions of diatom. Of course, the pigments for the yellow color is carotenoids. And they also do photosynthesis because of the green color pigments. Chlorophyll is also present. Okay. Now next one. So they are silicaceous cell wall, chief producer, diatomaceous earth used in polishing, filtration of oil and syrup. The next one, of course, you can see dinoflagellate. Where is the habitat of dinoflagellates? Habit of dinoflagellates. Yes, Dharani, very correct answer. Mostly marine. See, when we see diatom, these are the points we need to be very much remember because in cyanobacteria we discuss children fresh water and blooming on polluted water. Diatoms aquatic of freshwater and marine. But if you see protista, this one dinoflagellate mostly marine. Yes, mostly marine is the correct answer, Parthasarathi. And of course, they are photosynthetic because they have yellow color, green color, brown, blue, all these pigments are there. Also red. So these pigments are present in the cells. So here the outer cell wall is made up of cellulose only. Okay. Like there we discussed silica. Here we have cellulose. Why it is called dinoflagellate? Di means two. Number. Flagella. So they have a transverse and longitudinal flagella. So we have two flagella are present. So it uses the longitudinal flagella for locomotion, transverse for food gathering. So when it move like a top. So it is even called whirling whip. And you know very well children, it is having a color emitting characteristic feature. What is it called? Color emitting feature. The color emitting feature, emitting, is called, it emit colorful light. They produce light. And you know, red tide is also very good, Tilak. Very good. Excellent. Excellent. Yes, bioluminescence. And any of you remember the red tide is because of dash? Red tide. Very good, Dharani. Excellent. Yes, Gonia legs is the correct answer. Okay, this is possible question here. Gonia legs is for red tide. Of course, the sea appears red in color. Very good, Parthasarathi. That is the correct answer. Gonia legs. And can you see? It is having the structure. Of course, two flagella. One is longitudinal. Another one is transverse. It move like a top. I said it appears in different color: red, blue, yellow, green, and brown. And you can see the red tide also because of the gonia legs. And when it is more, it appears bright red color, looks very pretty, but it is toxin. Okay. And this toxin may affect the fishes. And from fishes, when the man consumes, it also affects the man and causes paralytic shell poisoning. Of course, it also reproduces asexually and sexually. So these are the possible questions from goniolics. The next one we are going to discuss is about euglena. What is the habit of euglena? Yes, very good, Shubhan. It is freshwater. So it is commonly found in freshwater. In both the previous case, we discussed they are aquatic, some are marine in chrysophyte, mostly marine in dinoflagellates. Here they are freshwater mainly. Okay. And euglena is not having cell wall. It is having. So in previous case, we discussed silica in diatom. Of course, cellulose in gonialex. Here it is pellicle. Okay, and this pellicle gives flexibility. Okay. And you know they are having two flagella. One is long flagella for locomotion. Another one is short flagella for creating whirlpool when it want to capture the food. As you can see a nucleus. Okay. And chloroplast also. When it is exposed to the light, it can prepare its own food autotrophic. When it is not in light, it behave like animal. As I said, it create a whirlpool using that smaller flagella, engulf the food. And you can find contractile vacuole, which is also a characteristic feature of animal. To balance the water, can you understand what is the role of contractile vacuole? Role of contractile vacuoles. Very good, Parthasarathi. Excellent answer. It is for asthma regulation only. So, contractile vacuole is for asthma regulation. Okay. And of course, the paramylum bodies where it reserve the starch. Okay. When it is exposed to the light, chloroplast, you can see they'll do photosynthesis. When it is not in light, it behave like animal. Of course, you know, it can create a whirlpool and engulf. Reproduction asexually, you know, fission it will undergo. And longitudinal fission. So you can see the structure of euglena. Paramylum storage device, you should know. Then, which is the unicellular saprophytic organism? Yes, yeast is also correct answer. Of course, very correct, Dhanani. Yeast is unicellular fungi, and in Protista, of course, it is slime mold. So, slime mold is the unicellular saprophytic organism. Of course, you can find it in wherever we used to say for the habit of fungi. You can find in decaying twigs, wood, logs, these areas, aquatic areas, and all you can find slime mold. Yes, Parthas are the very correct answer. So, these areas we can find slime molds. And during unfavorable condition, slime mold produce what? During unfavorable condition. Yes. What it produce during unfavorable condition? Yes, very good, Parthas are the fruiting bodies or spores is the correct answer. It produce spores. Do they have cell wall? Spores, do they have cell wall? Does the spore has cell wall? Yes, is the correct answer. Very good. Okay, so they have cell wall. Spores will have cell wall. Remember, contrastingly, the plasmodium does not have cell wall, no cell wall. So you should remember that during unfavorable conditions, spores have been produced. They have cell wall. Of course, when they spread and form aggregation, they are called plasmodium. So they will be exist in two forms. Of course, the spores, it may be in the form of mixed amoeba or swarm cell. Okay. So you can see the fruiting bodies, of course, spores, very correct, they have two walls. And you can see the fertilization here, the spores forms gametes. The gametes, here you can see the spores. The spores undergo and forms gametes. And the gametes undergo fertilization, zygote is formed. The zygote forms plasmodium. From plasmodium again, fruiting bodies that undergo meiosis and form spores, which are cell wall. Yes, are you all clear about this? Then protozoa, which are animal-like protista. So these animal-like protista are classified based on dash. They are classified based on their locomotory organelle. Am I correct? So it may be either pseudopodia, flagella, cilia, and non-locomotory are called sporozoa. Yes, sir? Fine. Let us see. So first one is amoeboid. Where do we find amoeba? Amoeba habit. So you can find, yes, moist region, very correct, Dharani. You can find it in water, aquatic in nature. Of course, the cell wall is made up of silica. Please remember, they are made up of silica. And of course, they locomote with the help of pseudopodia. And some are parasitic. And these parasitic example is entamoeba histolytica. So you can see the entamoeba histolytica here. And you know, amoeba reproduce through multiple fission also during certain condition. Only certain times it undergo multiple fission, unfavorable condition. Multiple fission is possible. Now this is flagellated, trypanosoma. Locomote through trypanosoma, flagella. So some are free living and some are parasitic. Of course, it causes sleeping sickness. So it affects the brain. And transmitter or vector is TC fly. So remember trypanosoma T for T. TC fly only transmits the trypanosoma problem. Of course, it is a flagellated protozoan. As it is flagella, only one is present. Then ciliates. So in cilia, organism, we have paramecia. So paramecia, what is unique when compared to other protozoa in terms of cell organelle? Anybody? So in terms of cell organelle, start with N. Something is unique here. Of course, nucleus. You can see two nucleus here. Micronucleus, macronucleus, possible question. So we have two nucleus present in paramecia. Okay. And how many number of cilia present? We have thousands of cilia present in paramecia. You can find a gullet here. Of course, it takes the engulf the nutrients via this only. Okay, holozoic mode. So micro and macronucleus, yes, one. So that is one possible question. Number of cilia, you should know 1000 cilia, and mostly freshwater organism only. So along with the water, food enters into the gullet. This is the gullet. They have contractile vacuole. You know that is for osmo regulation. So this is regarding ciliated protozoan. Yes, Parthasarathi. Very correct. Now sporozoans. See the sporozoans, they do not have any locomotory organelle. No locomotory organelle. And of course they undergo cycle like exoerythrocytic and endoerythrocytic. During that time, repeated mitotic division, multiple fission is possible. So multiple fission, which among the following, if the question is asked, amoeba, A option, B option is plasmodium, means we are supposed to take only plasmodium because it all the time undergoes amoeba only during favorable condition, sorry, unfavorable condition. Okay. And of course, as multiple fission is very important. And one more question. Most notorious for protozoan, most notorious. Which among the following? Is it amoeba? Is it flagellates? Is it ciliates or sporozoa? Which is most notorious protozoan? Most notorious protozoan is dash. Is it amoeba? Is it flagellate? Is it ciliate? Yes, Dharani, very correct. Excellent, Dharani. Sporozoan or most notorious. Plasmodium is a notorious protozoan. Okay. So we finished two kingdom. One is Protista, another one is Monera. Okay. Any doubt so far? I hope you are clear. I'll try to put the questions with reference to the biological classification and community questions also. Please try to solve as many as question possible. Try to solve assertion, reason, statement type questions and all. And shall we continue fungi in next class? So we can have another class for fungi and virus children. Thank you all for participating, answering sportively and motivating even the teacher. Thank you for your love and support, children. Thank you all. Any doubts, please stay back, ask your doubts, clarify and leave. No doubts? We can stop the class and resume back for fungi. So we can even proceed with important chapters, Subhan. Biological classification, we have 4 questions. So I am proceeded with it. Okay. As of plant kingdom, animal kingdom also important. Then we'll proceed to cell. Okay, Subhan. Or any other chapter you want, you suggest also. Yes. So some other day we can resume fungi and virus. Yes, Subhan. Yeah. Thank you. Thank you, Subhan. Thank you all children, those who are there.